This is my team consisting of Hui Hui, Isis, and me.
So today we'll be talking about the metabolic pathway of the three sweeteners, aspartame, xylitol, and fructose, and their various impact on health.
Sweetener is a substance that imparts sweetness to food. And now I'll be sharing about aspartame.
Aspartame is a non-nutritive sweetener, and it is composed of two amino acids, aspartic acid and phenylalanine, as well as a molecule of methanol. When aspartame is consumed, it is not absorbed into the bloodstream but digested by the enzymes in the intestinal tract. It is rapidly broken down in the small intestine into aspartic acid, phenylalanine, and methanol. Usually, phenylalanine is broken down into tyrosine by the enzyme phenylalanine hydroxylase. Methanol, the product of aspartame metabolism, is converted to formaldehyde by the enzyme alcohol dehydrogenase. Excess formaldehyde will be converted into formic acid, which is excreted in the urine or converted into water and carbon dioxide. Lastly, aspartic acid is involved in the synthesis of other amino acids, as well as the citric acid and urea cycle. Now I'll be talking about aspartame's impact on health. People with phenylketonuria, also known as PKU, are unable to metabolize phenylalanine into tyrosine as they lack the enzyme phenylalanine hydroxylase. And when people with PKU consumes excessive phenylalanine, it can cause detrimental effects such as intellectual disabilities, brain damage, and seizures. Now I'll be talking about my views on the use of this as part of our diet. Aspartame is generally recognized as safe to consume with over 200 studies supporting its safety. Aspartame contains the same amount of calories as table sugar. However, it is known to have a sweetness intensity 200 times more than sucrose. This decreases the amount of aspartame needed to achieve the desired sweetness level, which in turn reduces the calorie intake. Lastly, I'll be talking about the regulatory guidelines. The Food and Drug Administration, also known as FDA, established an acceptable daily intake for aspartame of 50 mg per kilogram of body weight per day. And the Singapore Food Agency requires any food that contains aspartame to include phenylketonurics, contain phenylalanine in the packaging.
Now I'll start on the metabolism of xylitol. Xylitol moves down the digestive tract after consumption. In the small intestine, absorption of xylitol happens. Approximately 50% of the consumed xylitol is absorbed through passive diffusion in the small intestine down the concentration gradient. The remaining 50% of the dietary xylitol thus enters the colon, where it can serve as an energy and carbon sources for the intestinal microbiota and leads to the formation of short-chain fatty acid, which provide energy to the host and support immune system homeostasis. Adsorbed xylitol is metabolized mainly in the liver, although kidneys and other tissues are also sites of xylitol metabolism. Also, red blood cells metabolize xylitol readily. In the liver, xylitol is first oxidized to xylulose by the enzyme xylitol dehydrogenase, causing the NADH to NAD ratio to increase. The formed xylulose is phosphorylated via xylulose kinase to xylulose 5-phosphate, an intermediate of the pentose phosphate pathway, which happens in the cytosol of the cell. Xylulose 5-phosphate is in the non-oxidative phase of the pentose phosphate pathway, where reactions are reversible. Xylulose 5-phosphate can either produce ribulose 5-phosphate, catalyzed by ribulose 5-phosphate 3-epimerase, or proceed on with the pentose phosphate pathway to form glyceraldehyde 3-phosphate and fructose 6-phosphate as the end products. Glyceraldehyde 3-phosphate and fructose 6-phosphate will then enter glycolysis to enable production of glucose. Now I talk about the impact that xylitol has on health. Xylitol has low glycemic index of 7 as compared to table sugar with a glycemic index of 60 to 70. Osmotic diarrhea may result if xylitol is taken in high quantities. It's a simple osmotic response to the presence of slowly absorbed carbohydrates in the gut lumen. The presence of this solute in the lumen will draw water from surrounding tissues. Xylitol also has benefit effects against mutant streptococci, commonly found on human oral cavity, which causes tooth decay. Xylitol is readily fermented by the intestinal microbiota, increasing formation of butyric acid beneficial to gut health. Now I talk about my views on the use of xylitol as part of our regular diet. Despite having the similar sweetness intensity, xylitol is a good substitute for table sugar due to it being non-carcinogenic, having low glycemic index, lower calorie count, as well as the benefit it brings to the colonic microbiota, which table sugar is not able to provide. This makes it suitable to be used in diabetic food and low-calorie confectionaries. In Singapore, we are fighting a war of diabetes, and having sugar replacement of sucrose with xylitol actually benefits people with diabetes. We can replace table sugar with xylitol partially or fully depending on the product that is being processed. For the example of cake, we can partially replace sucrose with xylitol as bulking and tenderizers are needed for cake. And also, helping to replace glucose with xylitol also helps in reduction of calories and helps in maintaining body weight, which reduces the chronic diseases that are related to overweight or overeating. And this helps in calorie control. For the regulatory guidelines, there is no specified guidelines yet. In Singapore Food Agency, additives may be added to the food at a quantity limited to the lowest possible level necessary to accomplish its desired effect.
Moving on, I'll be talking about the metabolism of fructose. Fructose metabolism involves three key enzymes, namely fructokinase, fructobiphosphate aldolase B, and thiokinase. These enzymes are only found in the liver and kidneys of humans. In the liver, fructose is rapidly converted to fructose 1-phosphate via fructokinase. Fructose 1-phosphate is then converted to triodihydroxyacetone phosphate (DHAP) and glyceraldehyde via aldolase B. DHAP is an intermediate metabolite in both the gluconeogenic and glycolytic pathway. As such, a portion of the original fructose carbon structure forms glucose. The glyceraldehyde is then phosphorylated by thiokinase to glyceraldehyde 3-phosphate, which is another intermediate in the glycolytic pathway. Up until this point, there hasn't been any rate-limiting steps, and as a result, there's an increased amount of substrate from thiophosphate, leading to metabolic pathway. Glyceraldehyde 3-phosphate is next metabolized to pyruvate via the rate-limiting enzyme pyruvate kinase. Fructose enhances the activation of pyruvate kinase and thus leads to an increased flux of pyruvate into the Krebs cycle. Next, I'll be talking about the impact of health of fructose. Fructose has a reduced glycemic effect as its metabolism does not promote any insulin response, therefore it is diabetic friendly. However, research suggests that fructose increases the risk of non-alcoholic liver disease and non-alcoholic steatohepatitis. To develop a fatty liver, it only takes at least 8 to 24 weeks on a high fructose diet with more progressive disease with longer exposure. Fructose has shown to induce other features of metabolic syndrome including elevated blood pressure, elevated serum triglyceride, and insulin resistance. Acutely, fructose stimulates thermogenesis and metabolic rate, but chronically, fructose shows a reduced resting energy expenditure, resulting in an easier weight gain. Next, I'll be talking about the view on the use of fructose as part of our regular diet. Fructose has become part of our lives since it is present in the majority of the food items we consume, such as fruits. However, evidence suggests that many substances present in natural fruits such as flavonoids, vitamin C, and many other antioxidants may protect against fructose-induced metabolic syndrome. Hence, the intake of natural fruits are not associated with non-alcoholic fatty liver disease. However, when fructose is used as a sweetener in our daily fruits such as in sucrose and high fructose corn syrup, it contains significantly higher amounts of fructose that are often ingested rapidly, leading to higher fructose concentration that will induce mitochondrial oxidative stress. In conclusion, although fructose has a reduced glycemic effect on humans, this pales in comparison to the drawback after consuming fructose for a long period of time. With Singapore declaring war on diabetes, fructose is not suitable as a replacement for table sugar.
This is the end of our sweetener presentation and we hope you like it. Thank you so much.
Thank
Thank you.